All right. So for the past paper revision lamai, api start karan.
Excuse me, teacher.
Yes, Ashita.
Teacher, like on 13th we have school, like we don't have holidays during this month. So on 13th we are having school till 9:30. If you can you have the class around 9:30?
At night.
No, like in the morning.
So from morning until 9:30, you have the class.
Yeah, like from 7:30 to 9:30 we have class at school. So are you possible to have the class after 9:30, teacher?
One second, let me check. I don't think after 9:30 will be possible. 13th. 13th, 14th, they come at 9:30 welding first. I have to do 10:30. Actually, after 9:30 is not possible. So let's do like this, Ashwita, Mayum, both. So third entire week you have extra classes at school. Okay, so what we'll do is for the maybe in a week take extra class and then third week take extra class fourth week take extra class right? So how we'll do this is that we now that we put the poll majority pull on the by any chance because practically it's not possible to get a slot that everybody can join, right? I know that having school and all that, I mean that's very important, but technically this is not a month, this is supposed to be your holiday month, right? So what we'll do is let's do like that, but for whatever you're missing, we will you can watch the recording, and for any doubts or for any clarifications you can talk to me. So let's do like that because I think that would be the most practical way. But you can personally contact me for any absentees who might not be able to join because of school or any other commitments. uh, uh, I didn't understand what you meant, Mayamdi. Last week before the starting of third term? What does that mean? You mean the third week or the fourth week of August that you have extra classes at school? So let me know that. So yeah, let's do like that. Let's try our best to do it like that way. And for any of you who might not be able to join because of school, that week only you have classes at school then, not before that. All right, okay, then no worries. It's a good, as I said, for any of those who are not, who might not be able to join the classes because of sorry, because you have school, I will be personally available for you for any doubts or clarifications, right? Let's do it that way. Okay, so for the past paper revision, since we are just starting out today, it's okay not to do it as homework. From next week onwards, we'll make sure that I'll make sure that I give you the questions to do beforehand during the class itself. So for the past papers, we are doing from 2016. So 2016, 17, 18, 19, 20, 21, then 22, 23 out of the, 23, 24 out of the, and 24, 25 out. Right? So, and 25, 26 out. So those are the, we have like 10 papers to do, 10 past papers to do. We have to be very familiar with the past papers, the structure, the questions, everything. අපි පටන් ගන්නේ, we will start with the 2020 paper. Right, 2020 paper එකේ නම් අපි ඉස්සරහට පස්සටයි move වෙනවා. So I'm not going to do it in the order. 2016 එකෙන් පටන් ගන්නව නෙවෙයි, but we'll start with 2020. Right? So there is a logical reason for me for starting from 2020, but you don't need to know that. Let's just start with 2020. Okay, I hope all of you have the paper. You have the papers, right? Am I not or the 2020 paper? Can you let me know now? Can you let me know now? Anybody who don't have the 2020 past paper? Huh? Everybody have, right? Then I will not bother sending the paper in the group. Huh? Past papers, we are first starting with the structured questions and the essay questions, and then much later only we are doing the MCQs. Okay. So in the 2020 paper, no, actually, yes. I did tell you about getting a separate book, right? Okay, so this is what you can do for because අපි දැන් theory pass paper දෙකම parallel යන හින්දා එකම පොතේ දෙකම කරොත් it's going to be very confusing for you. One option is take one book for theory and another book for past papers, right? Or the second option is you are doing the theory in the book no. Onna past paper questions එක පිටිපස්සේ නම් ඉස්සරහට කරගෙන යන්න පුළුවන්. Because sometimes, especially boys, I mean, මගේ experience එක ගොඩක් වෙලාවට questions ඒ කියන්නේ මේ පොත් නැති කරගන්නවා නිතරම. So එතකොට එහෙම misplace වෙන්න chance එකක් තියෙනවා. Just do it in one book. Theory ඉස්සරහා ඉඳන් කරන් යනවා නම් past papers පිටිපස්සේ ඉඳන් කරන් ආවත් කමක් නැහැ. But if you want to be a little more organized, you can take a different book. But if you don't have a different book today, you can manage with whatever you have for today. But next day onwards, you can do that, right? So we talked about the time allocation last week. To quickly remind you on that, I think you noted it down, no? Time allocation. Lama, can you all hear me?
Yes, teacher.
Okay. I just got a network issue. Let me share the whiteboard. All right. Okay. So yes, we were talking about the time allocation, right? Last week, our time allocation again, so we said for a structured essay question, generally it's 22 minutes, but you have to do within 15 minutes. And for an essay question, it's generally 30 minutes, but you have to do within 22 minutes. So 15 minutes, 22 minutes, right? So we are starting with 2020 paper. Please take it out for me. Take the 2020 paper real fast. Okay. It's raining here. I'm sorry, Lamai.
Tisha, you're lagging.
Yes, I think 1 second. It should be stable in a minute. I'm just changing to a different network. Is it okay now? Can you all hear me? And can you all see the board? Hari, it's raining a little heavily here. I'm in Colombo today, and I don't know if it's the same in your areas. But even though it was raining heavily, yesterday and day before yesterday, it wasn't raining that much. So it was not bad. But today it's raining. Anyways, so without further ado, without further talking, we'll get started. So one thing that I want you to do is please be honest to yourself, because this is not something that you do for me. This is something you do for yourself. Okay, I lost current also. Can you all still hear me?
Before it was lagging,
Okay.
now can hear Tisha.
All right. Thankfully, I'm connected to my hotspot, so I think it's all right. Okay. So let's start. In 2020, structured essay, that means paper 2, part A, right? The first question you cannot do. Api okum questions karana hai because api samhar lessons tam karna hai, so we'll skip those questions. So first question we'll skip. Go to the second question. Please be honest to yourself and do the question alone.
Tisha, we should do the second question.
Yes, I will also put the question to the whiteboard. Generally, it's 15 minutes, as we discussed. Mama, time ek set karana for around 12 minutes. And if you need a little bit more time, I'll give you until 15 minutes, right? Because some questions you can do a little bit early. Right. Okay, your time starts now. Lama, I'll keep my camera off because otherwise my laptop will run out of battery before we stop the class, before we finish the class. So just to save the battery, I'll keep my camera off. But I'm always here. I'm right here. Ask any question, put any question in the chat box. You can unmute, raise the hand, thumbs up, whatever. Okay? Okay, quickly do. I hope you're doing, Lamaya. If you have any questions, you can ask. I'll scroll down a bit. Lamaya, I will scroll down a bit. I hope you're still doing, huh? You have a little less than 1 minute. Lamai, I set the timer for 12 minutes. But if you want, I could give additional 3 minutes, but depending on if you want. So did you all finish? What's the situation? Anybody who needs more time? So everybody has finished? Okay. ඔගලන් කියන්න නැතුව මම දන්නේ නැහැ. So you have to communicate. Since none of you are communicating that you need more time, that means I'm assuming that you have finished. Okay. Now let's start the discussion. This is my favorite part. I'm just a little worried if current will stay out like this. Come on, discussion එකේදී මම camera on කරලා තියන්නම් otherwise. නැත්තම් discuss කරන අමාරුයිනේ මූණක් පේන්නේ නැතුව. Okay. So we are talking about organelles, right? Organelles we learned in the 6th lesson in grade 10. So there we talked about a few things, right? What did we talk about in that lesson? අපි මුලින්ම lesson එක කතා කරද්දී we talked about who observed the cells for the first time. Who observed cells for the first time. We'll just do a little bit of a revision of that lesson because that is how we do past paper revision. දැන් තව චූටි දෙයක් දැන් අද අපි ඔය revision revise කරන parts අපි ජීවිතේට ආයෙ revise කරන්නේ නැහැ. කියන්නේ මෙච්චර detail revise කරන්නේ නැහැ. So ඒකින්ද revision එකේදී හොඳට අහගෙන ඉන්න it's very very important. As I said that this is the game changer. Okay, so I got one answer. Who observed the cells for the first time?
Richard, Leiden, Swan.
No. Who's the person?
Isha, like who observed the cell for the first time, right? You're asking.
I'm asking who observed the cells for the first time. I got 3 answers out of 10 of y'all.
Isha?
Yes, Ashwita.
But before answer was wrong, I didn't understand the question. Shall I say my answer now?
Yes.
Robert who?
Okay, Ashwita says Robert Hooke. We'll see. Vindi, okay. I think someone else also unmuted, no? I didn't. I just very lightly heard a voice. Okay, Rashmika. Okay, yes, it is Robert Hooke. Robert Hooke observed the cells for the first time. Kiyan ba me MCQ wala haya? සාමාන්‍යයෙන් අර MCQ වල first questions දෙක තුන අහන්න ඕන කෙනෙකුට ලකුණු ගන්න පුළුවන් විදියට. So මේ වගේ question එකක් එතන අහන්න පුළුවන්. So ඒ වගේ simple ඒවා කරද්දන්න එපා, okay? Robert Hooke observed the cells for the first time when he was observing. He observed like a cork, right? A piece of cork, and what he saw was little beehive shape එකට තියෙන structures that are right. Beehive shaped structures. And he is the one who named them cells. එයා තමයි ඒකට cells කියලා name කරන්නේ. That was Robert Hooke. Okay? So මේ වගේ structured වල එහෙම අහන්නේ නැහැ. These are too basic, but they might ask in MCQs. Then who put forward the cell theory? Who put forward the cell theory?
Virtual Swan Sladen.
Okay, Ashwita. So something that one thing that we'll do is Ashwita and I think someone else also unmuted. How about we all put the answer in the chat box? Then I think everybody can because if somebody doesn't remember and somebody else says the answer out, maybe like we cannot test whether the other person remembers or not. So let's do that. Let's all put the answer in the chat box, or you can raise the hand and එතර unmute කරන කට්ටිය chat box එකේ දාන කට්ටිය වුණාට පස්සේ unmute කරන කට්ටියට උත්තර කියන්න පුළුවන් raise the hand කරලා. So ඒ වගේ මොකක් හරි දෙකෙන් එකක් කරමු. Okay? Okay, yes. So it was Schleiden, Schwann, and Radolf Virchow. Virchow, whatever. What was there in the cell theory, Aman? What was there in the cell theory?
Tisha, can you repeat the
What?
question?
Sorry, Ashita?
Can you repeat the question?
Yes. My question is, what are the points in the cell theory? Okay, Metni, I'll let you unmute. One second. Okay. Okay, Ashwita, also I'll let you unmute. Okay, Tinura, I got 1 point. Okay, Mayundi. Okay, Tinura, another point. Metna, okay. Rashmika, okay. Evindi, okay. Metna, okay. Okay, Metni, you can tell your answer.
The structural and the functional unit of an organism is a cell. Pre-existing cells form new cells. Each organism is made up of one or more cells.
Okay, Ashwita?
The structural and functional unit is cell.
Sorry, Lamai. Yes, go ahead.
And the existing cells are wait, wait.
So one is yes, as you said, all organisms. Sorry.
Tisha,
Yes.
all organisms are made up of one or more cells, and new cells are formed from pre-existing cells.
Yes. So the structural and the functional unit of life is cell. All organisms are made of one or more cells, and new cells arise from pre-existing cells, right? Or are formed from pre-existing cells. Make up what are these pre-existing cells? Are a mitosis, meiosis, swelling, then new cells. So that is cell theory. Then when you talk about the cell, there are we talked about two different structures: plant cell and animal cell. Okay. So my next question is, what are the differences between plant cells and animal cells? Again, we'll do in the same way as we did earlier. Differences between
You
plant
mean
cells
the
and
structural differences, right?
sorry?
The differences in the structure, like you asked the differences in the structure, right?
Yeah, yes. The organ. Now, please give me a second. Huh? Lamai, can you all hear me? I'm so sorry, the network is really bad. Let me share the screen again. Because I'm connected to my personal hotspot from the phone, it's not very stable. මට හිතෙන්නේ මම චුට්ටකට camera off කරලා තිබ්බොත් හොඳයි. I know it won't be the best for the discussion, but we have to be a little practical now, so I'll try to keep my camera off for a bit. If I feel like the connection is stable, I will switch it back on. So however, I did see your answers, right? So many of you have already put answers. Yes. So if you take a plant cell and if you take an animal cell, so plant cell, okay, you all said yes, there is a cell wall, right? Have a cell wall. Plant cell, okay, no cell wall. Okay. So animal cell, okay, no cell wall. Plant cell, okay, they have chloroplasts.
Animal cell, they don't have chloroplast.
Yes, animal cells, they don't have chloroplasts.
Large vacuole is absent.
Yes, in plant cell, large vacuole is present, animal cell is absent. Yes. Present here, large vacuole is absent. Small vacuoles may be there. So එක MCQs වල අහන්න පුළුවන් tricky ප්‍රශ්නයක් විදියට small vacuoles are present. What else?
More components are like cytoplasm.
Phytoplasm, what is that? Cytoplasm. Yeah. So in the plant cell, there was this large vacuole, right? I think camera on kana to amaru tika. Okay. So in the plant cell, there is a large vacuole, and in addition to the large vacuole, there's the mitochondria, there are Golgi bodies, there are chloroplasts, there are rough endoplasmic reticulum, smooth endoplasmic reticulum, all of that. So එතකොට ඔක්කොම organelles තියෙන හින්දා ඉඩරකම් cytoplasm එකට. Cytoplasm is pushed towards the periphery. Right? Cytoplasm එක, it's pushed towards the periphery. And yes, nucleus is also pushed towards the periphery. But in an animal cell, what's the case? Oh. I think now it should be okay. චුට්ටකට සමහරට disconnect වෙන්න පුළුවන් මම Okay, I got excited for no reason because power again went. Okay, anyways, yes. So as y'all were saying, in the plant cell, nucleus is pushed towards the periphery. How is it like in the animal cell? How is it like in the animal cell? Quickly.
What is the question you asked?
So, so we talked about three differences, right? So I'm talking about another difference. Some of you said the answer. In the plant cell, we said the nucleus and cytoplasm, everything is pushed towards the periphery, right? Push but in the animal cell, how is it different to the plant cell? Cytoplasm is pushed towards the periphery. But in animal cell, the cell is mostly yes. Yeah. Cell mostly contains cytoplasm. Because if you take an animal cell, animal cell can loku vacuole එකක් නැහැනේ. So there is more space. Right? So ekinda අර cytoplasm එක corner එකටම push වෙලා නැහැ හැමතැනම තියෙනවා. The plant cell is different. So these are the differences. Huh? So please keep these in mind. They might ask this for the exam. Okay. All right. So then we were talking about different organelles. Okay. So we first, yes, Mithmi.
Teacher, but doesn't the cytoplasm hold all the organelles?
Yes, it does. But so what they're trying to say is in a plant cell, now cytoplasm is a gel, right? So plant cell එකක content ගොඩාක් වැඩි හින්දා gel එක හරි චුට්ටි area එකක් තමයි තියෙන්නේ. ගොඩාක් corners වල තමයි cytoplasm වැඩිපුර තියෙන්නේ. If you might not have my note when we were doing the lesson, but in the textbook picture එකම තමයි මම හිතන්නේ මගේ note එකේ දැම්මේ. අර ලොකු වැකියාවල එකයි ඔක්කොම organelles තියෙන cytoplasm එක හරි චුට්ටයි cell එක මැද හරිය තියෙන්නේ. ඒතර ටික ඔක්කොම corner එකේ තමයි තියෙන්නේ. Right? Even though wherever there are gaps, cytoplasm is there. But gaps are very little. ඒකනේ cytoplasm පොඩ්ඩයි තියෙන්නේ. But animal cell එක there's plenty of space. So ඒ හැම space එකකම cytoplasm තමයි තියෙන්නේ. That's why mostly in the cell there is cytoplasm. Did that make sense with me?
Yes, teacher.
Okay. So then we have to talk about the organelles, right? So one of the organelles that we were talking about is nucleus. So you know, plant cell, animal cell, both of them have a nucleus. What is the function of a nucleus? What is the function of a nucleus? Okay, Tirura? Others? Okay, Mitmi? Okay, Rashmika? Okay, Mayundi? Okay, yes, so controlling all the life activities of the cell. So just like in a computer, all the controlling is done by the CPU, right? So just like that, oh, you can't see the whiteboard, 1 second. Evindi, I send the whiteboard link to the main chat. Huh? Click on it and see. Controlling all the life activities of the cell. So in a computer, you know, all the controlling is done by the CPU. If you take the cell, all the controlling is done by the nucleus. Also, so storing the genetic information. So you know, genetic information is stored in DNA. DNA are found in chromosomes. We have already done inheritance. Chromosomes are found in the nucleus, right? So storing the genetic information in the cell, and also, yes, transferring the genetic information. Because nucleus is what is responsible, because in mitosis, meiosis, all of that, cell එක දෙකට බෙදෙන්න කලින් nucleus එක තමයි මුලින්ම දෙකට බෙදෙන්නේ. ඊට පස්සේ තමයි cell එක ඉතුරු ටික දෙකට බෙදෙන්නේ, right? So that's very important. Then let's talk about, so nucleus is one of the organelles, right? Then let's talk about cell wall. Let's talk about cell wall. What are the functions of the cell wall?
Gives
Ashmita, raise hand, huh? Then I'll give you the chance. Okay. With me, Ashwita, I'll give you the chance. Mitna, okay. Mayundi, okay. Okay, what about others? Okay, yes. So cell wall එක mainly කරන්නේ it is maintaining the shape, right? So it supports the cell, supports and protects the cell, and it also maintains the shape of the cell. Another important thing about the cell wall is, Lamai, cell wall is a dead structure and it's made from cellulose. Made from cellulose and it's a dead structure. මේවා මතක නැති ඒවා මේ ඔයගොල්ලෝ note කරගන්න. I told you. Oh, I'm so sorry, Medmi and Ashwita. I couldn't call out your names. I'm very sorry. From next one onwards, I will when you raise the hand, I will allow you. Yes, Medmi.
Teacher, so they are asking the organelles in a cell and their main function. So there are about 2 to 3 functions, right? So should we write one?
If they ask you one, you write one. But sometimes they might ask you like 2 functions. But they might ask you to write 2 functions. Then you should know to write 2 functions. I mean, if that particular organelle has 2 functions, you should be able to write the 2 functions. Did that make sense with me?
Yes, teacher. So for nucleus, all these 3 answers are correct.
Yes. If they ask you 1, you can write one of these 3. If they ask you 2, you can write any 2 of these 3. Okay? All of these 3 are correct. Okay.
Okay, thank you, teacher.
So cell wall, okay. Cell wall supports and protects the cell, maintains the shape of the cell. The cell wall, okay, put the other features to it's made from cellulose and it's a dead structure. Okay, so remember that. So after the cell wall, we have the plasma membrane. So another thing about the cell wall, plants don't have, right? Animals don't have. Only plant cells have. Plasma membrane, of course, plant cells have, animal cells have. What are the functions of the plasma membrane? Okay, Mayumdi. Let me, Ashwita, I will give you the opportunity. Okay. For plasma membrane, you can't say bearing the organelles, not for plasma membrane. Okay, Metmi. Okay. Ashwita, you can tell your answer. Sorry, Ashwita?
It only absorbs materials like water, iron, vitamins inside the cell.
Yes. So yes, correct. Plasma membrane functions enclosed in the cell. Because if you take the animal cell doesn't have a cell wall, no. So plasma membrane is the one that encloses the cell. And other than that, it controls the entry and exit of materials. This is now going to kiwa nede lesson එක කරන කාලේ දැන් හිතන්න building එකක් තියෙනවා කියලා building එක වටේ තියෙන තාප්පේ වගේ තමයි cell wall එක. Cell wall is just there, it's a dead structure. නිකන් ගෙදරක තාප්පයක් building එකක තාප්පයක් ගත්තොත් it's dead, right? But plasma membrane is like the security guard. Plasma membrane තමයි බලන්නේ කවුද ඇතුළට එන්නේ කවුද එළියට යන්නේ මෙයා එයාව set නැත්නම් එයාව ඇතුළට ගන්නෙ නැහැ. සමහර අයට එළියට යන්න දෙන්නේ නැහැ, right? එයා තමයි හරි live structure එක. Right? එයා තමයි ඒක බලන්නේ. So this is made from, we said, proteins and phospholipids. Do you remember that? Plasma membranes, what are they made from? Yes, proteins and phospholipids. Okay. So, right. So that is about the plasma membrane, right? Now let's move on to cytoplasm. Let's do like this. Then we have discuss karna. I will give you like 5 minutes. Can you write the functions of these? So cytoplasm, mitochondria, chloroplast, Golgi body, rough endoplasmic reticulum, smooth endoplasmic reticulum. Okay, can you write the functions of these items? Mitochondria we put, Golgi. Haveka kapita ribosome, I forgot, no? Let's do this exercise. Okay, yes. And also backyard. I will give you like 5 minutes. Can you quickly write these? When a feature zone, just write the functions because that's quite important. That is what links this to the past paper question. Quickly do. Just 5 minutes, make it fast. Lamai, did you all write the function? Did you all write the functions, Lamai? Can you all hear me? Am I lagging? මං අහලා ගොඩක් වෙලා ගියාට පස්සේ නේ ඔයාලා respond කරන්නේ. Am I lagging or can you all clearly hear me?
Can hear clearly, teacher.
Okay, so you all write the functions. Okay. So now tell me, what are the functions? එකපාරට instantly හදිනේ ඔයගලන්ට පොඩ්ඩක් answers at එන්නේ නැත්තේ because you guys were not giving answers. That's why I'm giving you this chance. Okay. So what are the functions of the cytoplasm? What are the functions of the cytoplasm? Okay, okay, now I'm getting more answers, it seems like. Okay, Tinura. And okay, another answer. Okay, Mayundi. Okay, Metmi. Okay, Evindi. Metna. Okay, yes. So, Lamai, my camera off කරන් ඉන්නම්. මොකද දැන් battery best එකෙන් අපිට class එක දිගටම continue කරන්න බැරි වෙනවනේ ඒකයි. But if you want me to turn it on, let me know. I mean, if it's uncomfortable for you to have my like camera off, but otherwise let's do like this. Nothing practical, nanny. Okay. So maintain the shape of the cell. So it's something like this. Why do we say it maintains the shape of the cell? If you take a balloon, මොනාත් fill කරන්නේ නැති, air fill කරන්නේත් නැති, වතුර fill කරපුත් නැති balloon එකක් ගත්තොත්, එයා නිකන් අර like you know ඇකිලිලානේ, it's wrinkled, right? එහෙම තමයි අපේ cell එකත් if cytoplasm was not there. But balloon එකකට අපි air fill කරොත් හරි නැත්නම් අපි balloon එකකට ගිහලා water fill කරොත් හරි balloon එක හොඳට පිම්බිලා ඒ shape එකට හැදෙනවා. සමහර balloons දැන් ගොඩක් balloons රවුමට තියෙන. සමහර balloons තියෙන නිකන් අර පැපෝ එකක shape එකකට වගේ. ඊට පස්සේ ඔය bears ලගේ මූණවල shape එකකට balloons තියෙනවා. අපි එයා හරි water හරි දාලා fill කරාම තමයි අපිට shape එක පේන්නේ. So ඒ වගේ cytoplasm ඒ වගේ cell එකේ cytoplasm fill කරලා තියෙන හින්දා තමයි ඒකේ shape එක හරියට පේන්නේ. So that is very, very important. Okay. So maintains the shape of the cell. Then in addition to that, you guys also gave another use, bearing the cell organelles. This is something we kind of discussed earlier. All the cell organelles are embedded in the cytoplasm. And lastly, carrying out metabolic reactions. What do we mean by this? Is that there are certain metabolic means chemical reactions happening in the body, right? Our body එකේ chemical reactions what we call them metabolic reactions. So some other chemical reactions in the cytoplasm. So cytoplasm gives a medium for the chemical reactions to happen. So that is also quite important. Okay. What about mitochondria? Others? Yeah. So here what mitochondria does is production of energy or producing energy, releasing energy, whatever. That is why it's called the powerhouse of the cell. Either function don't write powerhouse of the cell. Because powerhouse of the cell is something that we just like call the mitochondria because of its function. Right? Powerhouse of the cell function you write the function. Okay. Then chloroplast what chloroplast? What do chloroplasts do? What do chloroplasts do, Lamai? You are very well aware of chloroplasts by now. Yeah, produces chlorophyll කියන්න එපා. Chloroplast එකෙන් chlorophyll හදනව නෙවෙයි, chloroplast එකේ chlorophyll තියෙනවා. So chloroplast වලට you can just say carrying out photosynthesis. Or you can say contains chlorophyll to carry out photosynthesis, or you can just say carrying out photosynthesis. Then we have Golgi body, right? What are the functions of the Golgi body? Yeah. Production of secretory substances. එක එක දැන් ওই සමහර cells වලින් ළමයි එක එකව secrete කරනවා. මොනවාද? For an example, in our respiratory tract there is a type of cell called එක ඔයගලන්ට syllabus එකේ නැති එකක්. මම නිකන් mention කරන්නම් අපේ respiratory tract එකේ nasal tract එකේ එහෙම තියෙනවා a type of cell called the goblet cells, right? Goblet cells. So goblet cells are the ones that secrete mucus. Then mucus is a secretion. So එතකොට මේ goblet cells ඇතුළේ ඉන්න Golgi complex එක නැත්නම් Golgi body එකෙන් තමයි ඒ mucus හදන්නේ, because mucus is secretory substance. So ඒ secretory substances හදනවා, ඒවා package කරනවා and ඒවා secrete කරනවා. Cell එකේ එළියට secrete කරනවා. So that is done by the Golgi body. It's very, very important. Then what about the rough endoplasmic reticulum? What about the rough endoplasmic reticulum? Endoplasmic reticulum there are ribosomes attached, if you remember, right? So what are the functions? Synthesizing lipids? No. Yeah. So the function is transportation of proteins inside the cell. So inside the cell, proteins are transported here and there by rough endoplasmic reticulum. Because you know, ribosomes වලින් තමයි proteins හදන්නේ. දැන් මම මෙතනම කිව්වා synthesis of proteins. The ribosomes produce the proteins and ribosomes are attached in the rough endoplasmic reticulum. So ribosomes වලින් හදන proteins ටික transport කරනවා rough endoplasmic reticulum, right? So that is what is done by the rough endoplasmic reticulum. Then what smooth endoplasmic reticulum does is what some of you mentioned earlier, synthesizing lipids, then steroids, and also transporting them in the cell. So yes, I got the answer as well. Synthesize karna kelame hadanama, okay? Synthesis of lipids and steroids and transporting them inside the cell, right? Then lastly, what about the vacuole? What are the functions of the vacuole? Okay. Maintaining water balance. What is maintaining water balance? Vacuole එක ඇතුළේ water තමයි fill කරලා තියෙන්නේ. Right? Plant cells are very rigid structures because inside the cytoplasm also has water, but plant cell එක ලොකුවට ඉන්නේ vacuole එකනේ. Vacuole එක අතර balloon එක වගේ වතුර මුතර පිරීලා තියෙන්නේ. So it maintains the water balance. And also it gives color. Yes, or provide color to the cell. So අපි සාමාන්‍යයෙන් ගත්තොත් එහෙම animal cells වල නම් අපිට වැඩි colors දකින්න අමාරුයි because we don't have a vacuole no. But in plant cells, දැන් ගොඩක් වෙලාවට cells අපි microscope එකෙන් බලද්දී we stain කංගේ පොඩි dye එකක් දාලා තමයි බලන්නේ. හැබැයි plant cells සමහර ඒවාට අපි dye එකක් දාන්න ඕනේ නැහැ. නිකම්ම උනත් microscope එකේ පේනවා. මොකද එක එක plant cells වලට එක එක colors තියෙනවා. Because vacuole has different pigments and these pigments have color. In addition to that, it also supports the cell. Okay. So these are the functions of the organelles. Now let's move to our question. I think Ibema we have discussed the question, right? So nucleus now for the function, you can write controlling life activities of the cell. This is just one function. Or you can write storing genetic information or transferring genetic information. ඒ වගේ මොකක් හරි answer එකක් ලිව්වා නම් correct. One of the three answers. Controlling the activities of the cell, controlling the metabolic reactions or metabolic functions of the cell, or like storing the genetic information, transferring genetic information, writing one of those is okay. My kakli work and okay, providing energy. What is this organelle? What is this organelle? Yeah, of course it is mitochondria. Mitochondria ke organ singular noun එක mitochondrian. නිකන් mitochondria ලිව්වත් කමක් නැහැ. මම නිකන් කිව්වේ. Then Golgi complex, what is the function? What is the function of the Golgi complex? Golgi complex, production of secretory substances, packaging and secretion. Right, that is the function of Golgi complex. I am good time spent because we just talked about these, right? Protein transport. Yeah. Rough endoplasmic reticulum. So I think that is clear for you, right? Does any of you have any questions? Does any of you have any questions? Is that all clear? Quickly, give me a thumbs up, everybody. It's really hot. I hope, do y'all have power? I don't know whether this is like something in our region only or is it like, do y'all have power? Okay. Hari, marks, tika mama denna marks. So if you wrote the right answer here, you get 1 mark. If you wrote the right answer here, 1 mark. Make a right answer, 1 mark. Make a 1 mark. Okay. So 1 for each 4 marks. Okay, Lama. So let's move on to the B part. An incomplete chart indicating the classification of plant tissues. All right. So this is quite interesting. Ma metana pe discussion karamu. So Lama discussion karanatare. I'm not asking you to write everything down. Ogan kamsina ඔක්කොම ලියා ගන්නත් පුළුවන් but ඕගලන් දන්නවනේ මේක මට මතකයි මේක මට මතක නෑ මේක මට ෂුවර් නෑ That is how the past paper revision is. You decide what you are going to write down. Okay. Okay. So let's talk about plant tissues. When we talk about plant tissues can be mainly divided into two. What are they? What are they?
Permanent tissue, meristematic.
Meristematic tissue
Politics, I
and
am really forgetting to raise my hand.
no, it's okay, it's okay. You are used to unmuting and speaking, so no worries at all. So meristematic tissues and yes, permanent tissues. So in meristematic tissues, a specialty ka mukadda, they don't have specialized functions. So they are all they can do is just dividing. Permanent tissues are divide when baha, but meristematic tissues can divide. And meristematic tissues don't have a specialized function. The only thing that they do is they are just dividing, right? So in meristematic tissues, they don't have any specialized functions. All they do is they can divide. But permanent tissues, they cannot divide, but they have specialized functions. In meristematic tissues, we talked about 3 types of meristematic tissues. What are they? What are they? What are the 3 types of meristematic tissues? Yeah, apical meristem, lateral meristem, intercalary meristem. Yes. Apical meristem ඔගන්න මතකයි shoot apex එකේ උඩම කොනේ තමයි තියෙන්නේ apical meristem. Lateral meristem තියෙන්නේ roots වල root apex එකේ. Right? So they keep dividing and adding more cells. එතකොට plant එක උස වැඩි වෙනවා. It increases the height of the plant. අයි උඩිනුත් root apex එක වැඩි වෙනවා. Root apex එකෙත් වැඩි වෙනවා. Lateral meristems are found parallel. Right? අරක මෙහෙම උඩ පැත්තටනේ lateral meristem එකේ මේ දෙපැත්තට. Right? So parallel to the plant, we have the lateral meristem. ඒකෙන් වෙන්නේ plant එකේ diameter එක වැඩි වෙනවා. Okay. Intercalary meristem තියෙන්නේ අර grass family එකේ කට්ටියගේ. Grass family එකේ අයගේ, so we have talked about this several times, I think. දැන් මෙහෙම කොලයක් හරි මොන හරි මේ nodes තියෙනවානේ. මේ nodes, these are called nodes. මේ nodes හරි තමයි intercalary meristems තියෙන්නේ. So they keep adding cells to the 2 sides of the node. එතකොට node එකේ තමයි වැඩි වෙන්නේ. This part is called the internode. Right? Nodes දෙකක් අතර gap එක internode. එතකොට nodes ළඟ තියෙන intercalary meristem, they keep adding more cells. So the length of this internode will increase. Internode එකේ length එක වැඩි වෙනවා කියන්නේ indirectly plant එකේ length එකත් වැඩි වෙනවා. That is intercalary meristem. Then is that clear, Lama? I'm going a little fast because these are like quite simple and it's a revision. But if there is any part that you find difficult or unfamiliar, let me know. Okay, then permanent tissues. No, length of the node. Intercalary meristem එකෙන් කරන්නේ node එකේ length එක. Not diameter. Diameter is like this, no? Length එක තමයි වැඩි කරන්නේ. Grass family එකේ තමයි තියෙන්නේ. Okay. So permanent tissues, then we have 2 types of permanent tissues. Simple permanent tissue and complex permanent tissues. So there were 3 types of simple permanent tissues that we talked about. What are they? What are the 3 types of simple permanent tissues? Yes, parenchyma. What are the functions of parenchyma? What are the functions of parenchyma? Parenchyma can be the most abundant tissue, right? Plant tissues are in godak mean parenchyma. Parenchyma is almost everywhere. It occurs everywhere. So, can you tell me some locations where parenchyma is existing? Can you tell me some locations where parenchyma is existing? Some okay. Yeah, cortex and pith of the stem, cortex and pith. If you don't remember cortex and pith, go to that particular lesson, that part, and check that, revise that. If you still don't understand, let me know. I will be happy to explain. So cortex and pith of the plant stem, cortex and pith of the root, right? And where else? Where else can we find parenchyma? Yeah, fleshy parts of fruit. Yes, correct. Seeds, yes, seeds actually in a part of endosperm killer. So that part contains parenchyma. Anywhere else? Leaves, correct. Leaves also contain parenchyma. Okay, so this discussion will talk and whatever the important parts will write. If you remember in parenchyma, we also talked about some features of parenchyma. What are the features of parenchyma tissue? What are the features of parenchyma tissue? Yeah. Others? Cells are spherical, or we called it isodimetric. That means there are spherical, cells. And also, yes, there are living cells. What else? I'm getting more answers, huh? Yeah. Intercellular spaces are present. Okay, what else? There's a thin cell wall and it's made from cellulose. Okay. Then what are the functions of parenchyma? Antakalama pictures balaganda, me plant cells pictures ogalangi ahanna puluwa. I think past papers karanga the Uganda penis. There were certain questions where they have given you images and they have asked you to identify the type of tissue. All right, so we were talking about yes functions. Photosynthesis. So in the plant leaf එකක් ගත්තොත් එමේ ඒකේ තියෙනවා two types of cells. එක type එකක් කියනවා අපි palisade කියලා. තව type එකක් අපි කියනවා sponge කියලා. Right? So palisade mesophiles කියලා ජාතියයි sponge mesophiles කියන ජාතියයි ඒ දෙගොල්ලන්ටම photosynthesis කරන්න පුළුවන්. And both of them are parenchyma cells. Okay, so photosynthesis is one of the most important functions that parenchyma do. What else? Yeah, food storage and water storage. Yes, water storage, correct. So, අපි ගත්තොත් එමේ potato වල එහෙම roots වලනේ හැදෙන්නේ potato yam එක. ඒ ඇවිල්ලා parenchyma tissue. Potato yam එක is parenchyma. Then if you take potato වල underground stem, right? Carrots වගේ ගත්තොත් එමේ it's and also sweet potato, it's the root. So in the root, food is stored. So අපි carrot වල කියලා okay, අපි carrot වල කියලා ගන්නෙ. Let's hope this time it stays. Let me quickly plug my device to the charger. All right, Lamai. Now I think you can finally see me properly. And yeah, great. So we were talking about yes, food storage. So it pass say roots wala food stock and the carrot wage api gatthoth theme of the carrot that we are eating, it's the parenchyma tissue. So actually potato ma uganda kalin kiyala tinawa meka potato plant එක එයාගේ underground stem එකේ food store කරගන්න ඒක මටම කවදා හරි ඕනේ වෙයි කියලා. අපිට කන්න දෙන්න නෙවෙයි. But we are of course digging it up and eating it, right? So yes, they store food in their parenchyma tissue. Water storage, if you take like aloe leaves, aloe vera කියන සාමාන්‍ය dry දැන් අපේ කොළඹ ඒ වගේ wet zone එකේ හැදුනට ගොඩක් එයා dry parts වල තමයි හැදෙන්න කැමති. හොඳට හැදෙන dry parts වල. Because aloe vera කියන plant එක හැදලා තියෙන්නේ dry parts වල වැවිලා they store their water inside the gel that we are taking for cosmetic purposes. It's their parenchyma tissue where they have stored water. So aloe, yes. Cactus, yes. Even a cactus, when you cut it across, you can see how they have stored water. Then there's a leaf called biofilm, right? Yes. So food storage, water storage, then photosynthesis. Anything else? Yeah, providing support, giving support. So some are herbaceous plants වල වගේ ළමයි ඒගොල්ලන්ට ලොකුවට stems එහෙම නැහැ. ඒතර ඒගොල්ලන්ගේ plants වල තියෙන parenchyma tissues වල ඒගොල්ලන් water absorb කරගන්නවා. Water absorb කරගත්තා මේ parenchyma tissues පිම්බෙනවානේ. පිම්බුණාම මේ plant එක නිකන් හොඳට turgid එකෙන් ටිකක් rigid, what do you call, like very sturdy විදිහට ඉන්නවා. Okay? So those are the features of parenchyma, don't forget. Collenchyma, what about collenchyma? What are the functions of collenchyma?
It is a living cell.
Yeah, functions. Yes, Ashwita.
It gives support.
Yeah. Others also, yes. And also photosynthesis, right? So support, you know, if you take dicot plants, you already know what dicot plants are. Egolangi, අපි දැන් look වෙනකොට එක එක stages තියෙනවානේ වගේ plants වල there are different stages. So okay, one second, something happened. It's okay, we'll continue. Okay. So all right, so collenchyma, yes. So if you take a dicot plant at a certain stage, they will start to form wood. When they start to form wood, plant right? But before the formation of wood, the plant is quite weak. So the collenchyma will support right? Collenchyma, and in addition to that, it also does photosynthesis. What about sclerenchyma? What about sclerenchyma? Okay, it's a dead cell. Ashwita, yes?
Provides support to plant body.
What else? I mean, functions wise, it's basically just giving support. Sclerenchyma, there were two types. One was sclerades, and then there was also sclerenchyma fibers. So they are both sclerenchyma. There are two parts, sclerades and sclerenchyma fibers, right? So keep that in mind. So if you take like sclerenchyma fibers, you can find them in like xylem tissue. You can find them in phloem tissue, like in coconut fibers. Because sclerades sclerate endocarp of coconut, and then the inside, what do you call it? The pericarp of mango. It passes like coffee wage seeds, date wage seeds, sclerades. Okay. So those are the pears fruit. Yes, that's correct, right? So these are where sclerenchyma is found, right? Okay, I think you got a rough idea, right? Now we're going to discuss කරද්දි තේරෙයි මේවා මට මතකයිද මේවා මට මතක නැද්ද? මේවා මට මතකයි කියලා හිතනවා නම් good. That means you have studied, so you are at a pretty good place. මේවා මට මතක නැහැ කියලා නම් ඔයගලන්ගේ idea එක, that means you have not studied and you should probably get started as soon as possible. එතකොට සාමාන්‍යයෙන් past paper revision එක කරද්දී the most important thing is you start to understand about yourself. You get a very good assessment. හොඳ realization එකක් එනවා මං ඉන්නේ කොතනද? What is my level? Where do I stand? What is my standard? එතකොට ඔයගලන්ගේ කියන්නේ ඔයගලා පාඩම් කරත් නැතත් ඒ සමහර lessons cover කරලා තිබ්බත් නැතත් that understanding where you stand is very, very important. That is the first step to getting an A grade. මුලින්ම ඔක්කොටම කලින්ම දැනගන්න එපා මම මොනවාද පාඩම් කරලා තියෙන්නේ මොනවාද පාඩම් කරලා නැත්තේ මගේ දැන් තියෙන සිටුේෂන් එක මොකක්ද කියලා දැනගන්න ඕනේ මුලින්ම. ඒක දැනගත්තට පස්සේ තමයි ඔයා strong areas you keep improving, weak areas you give more time to improve. That is how you do this, right? So past paper එකක් කරද්දී ඒ සමහර ඔයගොල්ලන් පාඩම් කරලා නැතුව you start to realize okay මේක මට මතක නැහැ මේක මම දන්නේ නැහැ කියලා. That is not a bad thing. That means you should probably study, right? Can you give me 1 second? Okay, Lamai. So complex permanent tissues, there's a bit of a power situation. We got power now, but in my house, in our living area only, we have another power. So that's why I'm still in the dark. But all good. So xylem and the other complex permanent tissue is phloem. Because I will be talking about xylem and phloem in detail in a different question, right? But just to sort of revise a little, in xylem tissue, it's a complex permanent tissue, right? That means it's made up of different types of cells. What are the 4 types of cells that you can find in the xylem tissue? Yes, Tenura. Okay, 1 second, Ashwita. Yeah, yes, others? Ashwita, you can also tell your answer now.
Tissue tracheids, parenchyma cells, fibers, xylem vessel element.
Yeah. So we have the xylem vessel element. We have tracheids. We have xylem
Excuse me,
parenchyma.
teacher.
Yes, Tashvita.
Is it a must to write in the orders?
Not really, not really. Order, that's why I just wrote in the order. But no, the order is not important. The 4
No,
parts are.
because I had this doubt before also to whether write in the order.
No. That's not important. Or maybe question the order is not important. But what is very important is, of course, knowing the 4 parts. You should also be able to identify the 4 parts in a diagram. In the textbook, you have given the diagram. Textbook एक diagram एक familiarize करेगा तो ये होंगे तो automatic. Okay, just familiarize the diagram on the textbook. I think even in the note of this lesson, mama picture kada dam mein textbook diagram එක बताना. Okay. So phloem में क्या मनाती है ने? Sieve tube element is there. What else? Companion cells. Parenchyma, and yes, good Lamai, phloem fibers. Okay, so again, I'm not going to be talking about how they appear, but because I told you, when a question है कि deeply revise करना, एक मैं इतना liquid discuss करने थे, but yeah, parenchyma एक कोतन गया, it's just parenchyma. May fibers पावा, even though मैं इतना specifically xylem fibers and phloem fibers के लिए they are still just parenchyma fibers. Just like that, parenchyma, this is xylem parenchyma, this is phloem parenchyma, they are both just parenchyma. Okay. Okay. So let's go back to our question. So plant tissues can be divided into 2. We already discussed this. Uda permanent tissues, the meristematic tissue. And then permanent tissues can be divided into 2, simple permanent, and now we already discussed it. Complex permanent tissues. Then simple permanent tissues, there's parenchyma, collenchyma, and what comes to C? What comes to C? Sclerenchyma. So complex permanent tissues, we have xylem, and what is D? Phloem tissue, right? Okay. Then what is the type of tissue in which photosynthesis occurs most? Now tell me, we already discussed this. No. Photosynthesis good amma not xylem. Yeah. No, not collenchyma. අනිත උත්තර දෙන්නෙම නෑ. එකම සෙට් එක තමයි උත්තර දෙන්නේ. What about others? Mayundi, yes, correct. Tinura, yes. What about others? Please give it a try. Whether you get the right answer or not. Photosynthesis occurs the most. The type of cell is parenchyma. Okay, it is parenchyma. Name the type of tissue which contains sieve tube elements.
Phloem tissue.
Kashita says phloem. Mayumdi, okay. Tinura, okay. Metna, okay. Yes, it is phloem. We just talked about this also, so I'm not going to take too much time again. So, mom, tissues gonna like I'm not talking much detail, but make up in the mama. We were talking about like simple permanent tissues more, but complex permanent tissues we'll discuss in a different question. But yes, so let's see how marks are given for meristematic tissues. For writing that, A 1 mark, B 1 mark, C 1 mark, D 1 mark. Then here for writing this 1 mark. I think for this also 1 mark. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Right. Now let's move on to the C part. Right. So this is photosynthesis. Did you all write this question? Did you all write this answer?
Yes, teacher.
Okay. So if you all remember, in photosynthesis, we did a few activities, right? We were discussing so what are the materials or ingredients needed for photosynthesis? Can you first tell me that? What are the ingredients, not ingredients per se, but more like raw materials? Or what are the substances needed for photosynthesis? Factors. Factors is the right word. What are the factors essential for photosynthesis? Okay,
Teacher,
Tinura?
shall I say?
1 second. Okay. All right. Others? Yes. Yes. So the factors essential for photosynthesis කියලා අපි ඉගෙන ගත්තා. We learned about first of all, we learned about carbon dioxide, right? Then we learned about water. Water is also an essential factor. And then we learned about sunlight and we learned about chlorophyll. So I have a small question for you all. Can you write the balance reaction for photosynthesis? Quickly write, 2 minutes, make it fast.
Teacher, you mean to type and send or write in the book?
Just write in the book, Ashwita. Quickly. Lama, did you all right? Balanda, make the live here, right? I'm not asking you for answers, but check. Can you all hear me? Yeah. So we know that for photosynthesis, we definitely need carbon dioxide. Carbon dioxide reacts with water. Carbon dioxide, where does the plant get? From the leaves, right? Through stomata. Water we are getting from the, so not we, sorry, the plants are getting from the roots, right? And in the presence of sunlight, that means it requires light energy. Light energy is obtained from sunlight. And nikang metun madi, chlorophyll must be there. So chlorophyll is only present in green plants, right? So chlorophyll also must be present. That is why photosynthesis only happens in green plants. Monad hadene glucose. Plus oxygen. Okay. So when we balance, I put 6 here, I put 6 here. I don't put anything here, but I put a 6 here. So this is how we balance this. Balance reaction for photosynthesis is this. Make a liwana hari, make a mokakar ekatana hari miss. So light energy, when you can write sunlight. Sunlight or light energy, it doesn't matter. But when a mokakari tanak verdina, you won't get marks. Okay. So carbon dioxide, manda meter again, common activities in detail describe karana, but do you all remember we did an activity to test for carbon dioxide where we selected a plant එක similar වගේ leaves දෙකක් select කරලා අපි දෙක polythene bags දෙකක දෙකකින් තද කරලා seal කරනවා. එක polythene bag එකට we just put water. To the other polythene bag we put KOH. Yes, correct, Mayundi. KOH. So KOH, what it does is it absorbs the carbon dioxide. So if you take the plant, so there's one leaf, there's another leaf. Make it, we just put water. Make it, we put KOH. So the thing with KOH is that KOH can absorb the carbon dioxide. එතකොට මොකද කරන්නේ මේ leaf එකට carbon dioxide එන්නේ නැහැ. Why? Because all carbon dioxide will absorbed by KOH. Okay? But this leaf gets carbon dioxide, water, sunlight, all of that. So it will do photosynthesis, but this will not do photosynthesis. Okay. Do you all remember that activity? Give me a thumbs up. Others? Others? If you all remember, I have a question. For this, we are taking a plant like this. අපි කියනවා අපි මේ activity කරන්න කලින් අපි ගන්නවා potted plant kept in dark for 48 hours. You can you all tell me the reason for this? Why do we take a potted plant kept in dark for 48 hours without just taking a plant? Why a potted plant? Okay, I got the answer from some of you. What about others? the answer from Metmi, Tinura, and Mayumti. What about others? Why do we take a potted plant that was kept in dark for 48 hours?
Shall I say?
Yeah.
As photosynthesis has occurred before, um, like glucose has formed and it has been stored as starch. To destroy the starch that is stored, the potted plant is kept in a dark room.
Okay. So I think technically the answer is correct, but one word that you use, destroy, word that you use, you might not get marks. The thing is because for the plant, they keep storing starch. Can have a glucose because starch with store right? When they are kept in sunlight, they keep making starch and also, of course, in the body, cellular respiration, well, the glucose use karana. Photosynthesis welling hadana, cellular respiration use karana. Ehema tamai normally when. But now it's kept in dark now. So photosynthesis karenne. What does cellular respiration use? All the stored starch. Store karapu starch to okkum cellular respiration will use karama. All the stored starch will be over. After 48 hours, that means 2 days, this potted plant will have no stored starch. Because starch test if you take this one, photosynthesis when right? And this is not supposed to give a color change for the starch test. Or a stored starch, this might give a color. Right? Even though a පැය 3-4 starch හදන්න නැහැ මොකද photosynthesis වෙන්නේ නැහැ නේ KOH is absorbing all the carbon dioxide. But if it has stored starch, it will give a positive result. That is why we are taking a potted plant kept in dark for 48 hours, right? Then what was the activity we did to test for light energy? If you all remember, we took a leaf or 2 leaves or 3 leaves or how many black color color sorry, we took 2 leaves, right? We took 2 leaves. I just forgot for a second. Black color polythene hacking cover colorless polythene like lunch sheet polythene hacking cover and then we kept it under sunlight. Again, we are taking a potted plant kept in dark for 48 hours. මේ black color polythene එකේ cover කරපු carbon dioxide උත් තියෙනවා, water තියෙනවා, chlorophyll උත් තියෙනවා, but it's not getting light energy because black color doesn't allow sunlight to pass through. So ඒතර මෙයා photosynthesis කරන්නේ නැහැ, මෙයා photosynthesis කරනවා. ඒතර මෙයා starch test එකේ color එක දෙන්නේ නැහැ, මෙයා color එක දෙනවා. Okay, we are taking again a potted plant kept in dark for 48 hours. Then to test for chlorophyll, what we used was a mosaic leaf. ඔගාන මතක දන්න අපි නිකන් mosaic වගේ pattern එකක් තියෙන leaf එකක් ගත්තා. මුලින්ම මේ leaf එක අපි සුදු පාට කොලයක trace කරගන්නවා. Yes, like a croton plant leaf, right? First we trace it because otherwise මෙයාගේ අපි starch test එකේදී ඔගාන මතකයි usually how we do starch test is we first boil the leaf in water and then we take it out and then we boil it in an alcohol in a water bath. එහෙමනේ අපි starch test එක කරන්නේ. ඒතර මෙයාගේ colors එක එක තැන් වල තමයි පෙන්වන්නේ. එතකොට අපිට අර කලින් තිබ්බ pattern එක මතක නැත්නම් we'll be confused. So you take a leaf, you trace the pattern of the leaf on a piece of paper first. ඊළඟට ඔයා මේකට starch test එක කරනවා. So let's say මේ තමයි green color තිබ්බ areas කියලා. Green color තිබ්බ areas වල chlorophyll තියෙන හින්දා those areas do photosynthesis, but let's say maybe red color areas here, the red color areas will not do photosynthesis because red color means chlorophyll net. Okay, that is how we tested for chlorophyll. I have a question. Of course it's wrong. So, okay, this is a good question. It was asked in an MCQ, which was hard to test among these tests. So, අපේ මේ factors ටික ගත්තාම our factors are the 4 factors that are essential: carbon dioxide, sunlight, chlorophyll, and H2O. මෙතන මට හරියට 2 එක හැම type කරන්න බෑ, but excuse me for that. Carbon dioxide, no problem at all. We can test no. We can test කරන්න පුළුවන්. Sunlight again no problem at all. අපිට මේ test එක කරන්න පුළුවන්. Chlorophyll also no problem because we can take a croton leaf and we can do the test. Water තමයි අපිට test කරන්න අමාරු. Why? Because now if you take carbon dioxide test, what we did was එක්කෙනෙකුට carbon dioxide දීලා අනිත් එක්කෙනාට carbon dioxide දුන්නේ නැහැ. KOH දැම්ම කියන carbon dioxide එක absorb කරාම it didn't get carbon dioxide. So අපිට මොකක් එකක් test කරන්න ඕන නම් යාව දීලත් බලන්න නැති වුණාම බලන්න. Here when I want to test for sunlight දීලත් බලනවා sunlight දෙන්නේ නැතුවත් බලනවා. When I want to test for chlorophyll මේ parts වල chlorophyll තියෙනවා මේ parts වල chlorophyll නැහැ. But water වල මට එහෙම දෙකක් කරන්න බැහැ. Water තියෙන plant එකයි water නැති plant එකයි. එහෙම කරන්න බෑ. Because the moment the plant doesn't receive water it will just die. Photosynthesis කරන්නත් මුලින්ම it has to be alive. So therefore, the factor that we cannot test is water. Okay. All right. So factors are carbon dioxide, water, chlorophyll, and sunlight. Hadana products manada. This is glucose, anitha kena oxygen. Glucose test අපි already, how do we test if glucose was produced? So in photosynthesis, we know glucose is produced, right? Oxygen is a byproduct. Yes. In photosynthesis, we know glucose is produced, right? හැබැයි මේ glucose ටික store වෙන්නේ starch විදියට. එතකොට photosynthesis වල glucose හැදුනා කියලා අපිට මේ leaf දැන් හිතන්න දැන් මේ leaf එකේ මෙතන මේ C6H12O6 is glucose. This is glucose. But just because glucose was formed, I cannot go and do a glucose test. Because glucose haduna hatiye, the glucose will be stored as starch. So I cannot do a glucose test. What I have to do is, I have to do a starch test. So in the carbon dioxide test, in the carbon dioxide activity, in the sunlight activity, in the chlorophyll activity, in all these activities, we did the starch test. No, the starch test kiyala karanne mokadda. Itana me leaf එක කියලා. First, I boil this leaf in water. ඔගන්නේ මතක ඇති steps ටික, right? I boil this leaf in water. When I boil the leaf in water, what happens? When I boil this in water, what happens? Why do I boil the leaf in water?
To remove the chemical.
No. Yeah. It's Mayundi, that's correct.
Sorry, teacher.
Okay.
Like.
Yes, go ahead.
Give me a second, teacher. Like, I'm coming to say, but like.
No worries. Yes, okay. So, met me, okay. Okay, so මෙහෙමනේ පොඩි ප්‍රශ්නයක් තියෙනවා. අපි stored starch නේ දැන් okay. Photosynthesis හදන්නේ glucose තමයි. I told you glucose හැදුවට glucose test එක කරලා වැඩක් නැහැ. මොකද හැදෙන glucose ඔක්කොම starch විදිහට store වෙනවා. Right? So starch විදිහට store වෙනවා කියන්නේ we have to do the starch test. Do you remember in the starch test what happens is if there is starch, I put iodine and it will turn to a dark blue or like a blackish blue or a purplish blue, right? And podi ප්‍රශ්නයක් තියෙනවා leaf එක already green color නේ. Green color leaf එකට මම iodine දැම්ම කියලා මට ඔක හරියට පේන්නේ නැහැ. එතකොට මම ඔක්කොටම කලින් කරන්න ඕනේ මේ leaf එකේ green color එක අයින් කරගන්න එක. ඒකට තමයි මම මේ steps ටික කරන්නේ. Why is leaf green in color? Because of what? What is the pigment giving the green color to a leaf? Chlorophyll. Green color එක අයින් කරන්න මම මුලින්ම chlorophyll අයින් කරන්න ඕනේ. Chlorophyll is inside the cells. So chlorophyll අයින් කරන්න කලින් මම මුලින්ම cell එක you know, always a cell is blocked by the cell wall. Cell wall එකෙන් කරන්නේ cell එක protect කරන එකනේ. මුලින්ම cell wall එක weak කරගන්න. Weaken or breaking the cell wall, right? Cell wall එක break කරාට පස්සේ දැන් chlorophyll ටික එළියට ගන්න ලේසි. ඊළඟට මම මේ leaf එක alcohol ටිකක දාලා water bath එකක heat කරනවා. Why do I boil in alcohol? Why do I boil in alcohol? Yeah. Yes, ma'am, the others. I don't know what others are doing. Did you all study? Did you all even do the question? Don't be lazy, huh? අපි alcohol වල boil කරන්නේ එතකොට අර chlorophyll ටික ඔක්කොම එළියට ඇවිල්ලා alcohol වල dissolve වෙනවා. So the thing is ළමයි මම මේ step එක කරේ නැත්නම් මෙතන chlorophyll ටික එළියට ගන්න බෑ මට. Because cell wall එක හරි rigid. Yeah, chlorophyll ටික block කරන් ඉන්නේ. But when I first boil in water, now the cell wall is weakened. Then when I boil it in alcohol, I can take the chlorophyll out, right? So I'm boiling this in alcohol to sorry, Lamai. Let me share the screen. I look like a ghost, no, in the dark. Light again, or tawat ghost again. Let me check if my phone has battery. I'm just gonna take a little bit of your time, huh? Okay. So we boil this in alcohol, and the next question is, why do we boil this in a water bath? I mean, water. අපිට මට කෙලිම alcohol එක පුළුවන් මේ Bunsen burner එකෙන් heat කරන්න. Why do I have to use a water bath? Huh? Why a water bath? Because alcohol is highly inflammable. That means even with a small spark, alcohol can catch fire. Right, that is why. So this was then after that we take the leaf out. The leaf will not have a green color, right? So yeah, inflammable it catches fire. Yes. Okay. So okay, sorry, Lama, it's a little dark. Okay, anyways. So that is why we use a water bath. And then, as I said, now the leaf will not have its green color. එතකොට අපිට starch test කරන්න හරියම lazy, right? Then you just put iodine, and if it has starch, it will give like a blue-black color. If it doesn't have starch, it will just give the same iodine color. Mind, I didn't get the question that you're asking. Is it clear? Oh, you mean inflammable, flammable, same word. If that's what you're asking, I'm not sure. If that's what you're asking, inflammable what, flammable what, it's okay. It's the same. Right. Okay, yeah. It's the same thing. Inflammable and flammable opposite way, so don't worry about it. They're both the same word. We just do the starch test. So that was for starch. The other product means the byproduct is oxygen. Oxygen වලටත් අපිට වෙනම test එකක් තියෙනවා. What is the test? That is exactly what they're asking in this question. So we'll straight away jump to the question. If you look at the question, we have hydrilla plants and they're covered with a glass funnel. And there is a test tube, there is water and all that. Okay. An apparatus set up by a group of students to investigate a product of photosynthesis is shown in the diagram. What is the gas collected in the test tube when this apparatus is kept in sunlight? Sunlight වලදී වම photosynthesis වෙන oxygen තමයි හැදෙන්නේ. So the oxygen gas produced will be collected at the top here, right? So here the gas collected is oxygen. නිකන් oxygen නැතුව නිකන් O2 කියලා ලිව්වත් කමක් නැහැ. Okay. State a test that can be done to identify the gas and the observation you make during that. What kind of a test can you use to identify oxygen? Okay. Others? Glowing splinter test. Yeah, කමක් නැහැ. I mean, glowing splinter test කියලා test එකක් අපි හැමෝම ඉගෙන ගත්ත නෑ, but you can just say inserting a glowing splinter into the test tube. Right? Then what happens? What is the observation? It reignites with a flame. Test is glowing splinter test කෙනෙකුට ලකුණු දෙයි, don't worry. Inserting a glowing splinter into the test tube, and observation is it will reignite with a flame. It will reignite or relight with a flame. That's the observation, right? It burns or something like that. A new apparatus similar to the above apparatus was made by putting water saturated with carbon dioxide instead of normal water. Okay. What is meant by saturated? What is meant by saturated? Nikang with කියන එක නෙවෙයි. What is saturated? Inserted. No. Well mixed or absorbed. No. Forcefully, no. This is something you know already, yeah? What are the other answers that you all have? No, not dissolved. Yeah, dissolved tamai, but that's not exactly what saturated means. Saturated kiyanne lamai, saturated with carbon dioxide කියලා කියන්නේ ඒ solution එකේ ඒ item එකෙන් ඊට වඩා dissolve කරන්න බෑ. For an example, let's say you take like 100 grams of water. ඔයා මේකට චුට්ට sugar dissolve කරනවා. පොඩ දදා dissolve කරනවා. එහෙම දානකොට there will come a point that ඔයා add කරන sugar එක dissolve වෙන්නේ නැතුව මෙතන යට precipitate වෙනවා. ඒ කියන්නේ මෙයා saturate වෙලා. Saturated කියලා කියන්නේ a volume එකේ a solvent එකේ in that particular volume the maximum amount of solute is dissolved. A solute එක maximum amount එක dissolve වෙලා තියෙන. ඊට වඩා dissolve කරන්න බෑ. If you take this water saturated with carbon dioxide, What that means is the maximum amount of carbon dioxide is mixed in the water. So it's saturated, we call it, right? So normal water gun, water saturated with carbon dioxide gun. So if you take normal water and water saturated with carbon dioxide, what is the difference? What is the difference between these two? Normal water will carbon dioxide put right? Less carbon dioxide. Yes, but water saturated with carbon dioxide has much more carbon dioxide. So this is the difference, right? So keeping that difference in mind, let's go to the question. So, අපි දැන් කලින් apparatus එක use කරලා තියෙන්නේ just water. But here we are using water saturated with carbon dioxide. State an observation that could be expected with regard to the evolution of gas bubbles in the new apparatus when comparing with the evolution of gas bubbles in the first apparatus. Okay. Okay. Mayumdi, Thinura, I got your answers. Others? Yes. So, බලන්න අපි photosynthesis equation එකට ගියොත් normal water වල carbon dioxide. So let me just erase this. Normal water වල carbon dioxide පොඩ්ඩයි තියෙන්නේ. Right? Carbon dioxide පොඩ්ඩක් තියෙන හින්දා හැදෙන oxygen උත් පොඩ්ඩයි. But if you take water saturated with carbon dioxide, there is more carbon. More carbon කියලා කියන්නේ automatically there is more oxygen produced. Carbon dioxide වැඩි කියන්නේ reaction එක වැඩියෙන් වෙනවා. Oxygen වැඩිපුර හැදෙනවා. So අපේ ගන්න observation එකනේ oxygen gas වැඩිපුර හැදෙනවා කියන්නේ gas bubbles evolve more, right? So you can say gas bubbles evolve more. The rate of evolution of gas bubbles is high. Or gas bubbles evolve with a faster rate or higher rate. Yeah. Give reasons for the observation you mentioned above. I already got one answer. I got another answer. Echo කියන්න පුළුවන්. That කතාව හිටන්නේ so carbon dioxide වැඩි කියන්නේ carbon dioxide is a reactant no in photosynthesis. Carbon dioxide. So basically what you have to say is there is more carbon dioxide. ඒ කියන්නේ photosynthesis, the rate of photosynthesis is also high. Rate of photosynthesis high නම් photosynthesis වල product oxygen. Therefore oxygen will also be produced more. That's the reason. ඔයාගේ reason එක flow එකක් තියෙන්න ඕනේ. Right, so you can say since there is more carbon dioxide, rate of photosynthesis is higher. Then more oxygen gas will be produced. This is what you have to write. මේ වගේ මීනිය එක තියෙන විදිහට ආන්සර් කරලා තියනවා නම් you get marks. Is this clear, Lamai, up to this point? I'll tell you how marks are given. With an oxygen කියලා 1 mark. Here for the test, inserting a glowing splint into test tube 1 mark. It will reignite, relight, reburn with a flame cure 1 mark. For this 1 mark, one second. Here also 1. So very clear in this one. Yeah, 1, 1, 1. 1, 2, 3, 4, 5, 6. Yeah. So for the second question, can you all tell me out of 15, how many marks did you all get? Okay, Mayumdi, I got your marks. Good. Thinura, okay. Metna, okay. Savinu, okay. Others? Metmi, all right. Anyone else? Okay, so that's pretty good. So today we were not able to discuss lessons, but in the extra classes, we will discuss the lessons, right? Can whiteboard note එකක් එවන්න ළමයි. Can you all mark these questions for homework? අද අපි පොඩක් වෙලාව ගියා මොකද ක්ලාස් එක වෙලේ questions උත් කරන්න උනානේ. But this week we'll mark the questions for homework. 2000, this is 20 paper, right? 2020, okay. Third question. Paper 2, huh? This is also, it's a chemistry question. So at home also be honest to yourself. Don't look at the marking scheme when writing answers. And also among kiyadi pidira time එක දාන්න. Right? Third question you do. Fourth question also you do. Let's also do the fifth question. Fifth question is in the essay part. Huh? I think this is enough. In paper 2020, paper 2, question 3, which is a chemistry question, 4 is a physics question, and 5 is a bio question. This is your homework for next week. Is this clear to you, Ramai? mark all good, right? Okay. Do you all have any questions today or is everything clear? Give me a thumbs up if everything is clear. Mayundi, okay? This. Is everything we did today clear? Metmi, okay? Metna, okay? Tinura, all right. Rashmika, Savinu, Vihan. All right. So, mark the homework. We'll do these 3 questions. Let's see how you respond to the poll. And depending on that, either 13th or 14th morning, which is Thursday and Friday, we'll arrange an extra class where we can finish electrochemistry. Extra classes will be papers, we are only doing theory. Huh? So ඒක අපි ඔයගලන්ගේ response එකත් එක්ක arrange කරගමු. Then I think we can wrap up, right? Let's wrap up and let's see each other on 15th of August. 15th of August. Okay, all good, right? Okay, let's wrap up, Lamai. And I'm sorry for the disturbances today because it was constant power cuts, power coming, going, and like that. But at least we were able to do the class until the very end, for which I'm quite happy. Yeah, rest of it we'll discuss next week. Okay, Lamai. Thank you so much and good night. Until we meet next, have a great week, Lamai.